Okay. Thank you very much. I think a good afternoon to all of you, and also from our WESTPAC countries. So great to see you. So on behalf of the IOC Subcommission for the Western Pacific, IOC-WESTPAC, I would extend our sincere welcome to all of you for this WESTPAC webinar series on ocean acidification. So as part of the UN competent agency for ocean scientific research, I think IOC-WESTPAC, as you are aware, has been playing an essential role in promoting ocean acidification research and cooperation in the region. So since 2014, a lot of efforts has been made in the region in networking, conducting the regular scientific workshop, and developing research capacity, and also developing standard operating procedure for ocean acidification monitoring. So after several years' efforts, I believe you can also say, a very increasing impact of the ocean acidification program that has been yielded over the last 5 years. So which has been demonstrated by, the number of the researchers and the institutions in the region engaged in the network and also engaged in the ocean acidification network. And also you can see a very increasing number of monitoring stations in this region. If you can recall our first meeting in Phuket 2015, during that time, there were only around, 30 pilot station that has been identified 5 years ago. But now I think the number increased to more than 150 stations based on the survey we have conducted last year. So we sent a questionnaire to all network members and to understand what the current monitoring status is in different countries. So I'm very pleased to say the impact has been gradually generated through our joint efforts over the last 5 years. But of course, this is far from enough. So to overcome this kind of pandemic, so this ocean acidification webinar aims to share the latest ocean acidification knowledge generated among our network research institutions in this region, and also identify the good practice. I think many researchers are still going on at different level and in different areas. So we very much want to identify what are good practice that we can replicate about ocean acidification research and monitoring. But more importantly, to brainstorm a way forward, particularly in the UN Decade of Ocean Science. That means whether we can move one step forward to inform decision, to inform conservation, and also inform policy through the consolidated knowledge and service, with their strong collaboration among all institutions in the region. So I'm very glad to see, there are also several young generation ocean professionals who also take an active part in this ocean acidification webinar series. I sincerely hope you will keep engaged in and continuously developing your leadership through the interaction with our senior scientists and professionals from this region. So I want to send my special thanks to our senior professional and also scientists from this region, particularly Somgat from Thailand, Eileen from Malaysia, Melo from the Philippines, and also Li Yang from China, and also Apple from Thailand, for your passion and also enthusiasm for promoting the ocean acidification research and cooperation among all research institutions in this region. So I do hope through the webinar series, you could have a very wonderful and joyful discussion. as I say, not only to share the latest scientific knowledge, but also brainstorm the way forward for our future ocean scientific research in this region, and also for an enhanced impact of the scientific knowledge for decision and also policy. So with that, I would like to hand over the floor to Somgat and also invite him to moderate the Foster Ocean Education webinars today.
Yeah.
Somgat, floor is yours.
Yeah, thank you very much, Wenxi, for your very nice, openings, remarks. I think it's today we are going to have the speaker. It's as you see on the screen already. I think it's since the time we have about two hours, and each presentation for 3 presentations will be take each a half hour per each of those presentation. It totally will be 1.5, and then we will have a half hour for the question and answer at the end of the 3 talk. I will not break between each talk with the question. We will go to the end. You can wave your hand on the screen, and then I can let you ask the question later on. Yes, I think I would like first I would like to invite the first speakers, Dr. Liang Chang. He is a research fellow at the key laboratory of the Global Chain and Marine Atmospheric Chemistry, 3rd Institute of Oceanography, Xiamen, China. He has long experience work in the field of nitrate study in the Arctic and Southern Ocean. During several years, Dr. Liang and his team has been carrying out his research on the carbonate chemistry in the coastal water, including develop equipment for monitoring of the carbonate parameter in the seawater. As he's going to give the presentation today on the underways measurement of dissolved inorganic carbon in the estuarine water. Yeah, Dr. Liang, it belongs to you now, please.
Okay. Can everyone hear me?
Yes.
Okay. Yeah.
Okay. So yeah. So here we go. Okay. Good afternoon, everyone, and yeah, I think it's my honor to participate in this webinar in my presentation on this underway measurement technique. And Liang Zhan from Key Laboratory of Global Change and Marine Atmosphere Chemistry, Ministry of Natural Resources, and our institute is based in Xiamen. And this work also, we have a collaborator from University of Malaysia, Terengganu, Dr. Xingqi Po, and he is also participating in this webinar. Okay, so though this presentation is about the technique, but I will give some introduction about the background of our study and introduction of what we are doing now. And of course, is the case study of these dissolved inorganic carbon measurement methods. And I will finally talk about the future study plans. Okay, so as everyone here knows that we are now under the impact of climate change or global warming. And we know the cause is the anthropogenic emission of carbon dioxide by industrial fossil fuel combustion, and carbon dioxide concentration in the atmosphere is keep rising. So besides, we know that not only the carbon dioxide is a greenhouse gases, there are also other greenhouse gases such as methane and nitrous oxide, which is also increased rapidly in the atmosphere. And though these 2 gases is not so, the concentration as these 2 gases are not so high as that of carbon dioxide, but their greenhouse effect is higher than carbon dioxide on molecular basis. So the three greenhouse gases is very important, and we know that because of this greenhouse gases emission, the 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 climate change is the global temperatures increasing. So the ocean is plays a very important role in regulate the global climate, and it absorbing about 1/3 of anthropogenic carbon dioxide, which relieve the global warming somehow. But meanwhile, because it's absorbed too much uh, carbon dioxide and its pH value had already dropped zero point 0.1 unit since industrial revolution. So this now is already a very serious crisis for the ocean ecosystem. Moreover, we know that because of global warming, the ocean is also the temperatures ocean is also raised. That that also decrease the solubilities of the ocean, the solubility of oxygen in the ocean, and contribute to the phenomenon called the oxygenation. So since 1960, there's about 2% of global ocean oxygen inventory lost. So it's also a very serious um, issue in the coming years, we think. And in the coastal region, the um, yeah, I think the human impact is more serious, more directly. We know that the agriculture and livestock runoff and residue runoff and urban runoff, they carry large amount of sorry, they carry large amounts of nutrient and discharging to the river and the estuary and the coastal ocean, which result in the eutrophication. During these processes, um, there's a lot of organic matter produced, and when this organic matter mineralized, it consumes large amount of oxygen and result in hypoxia. So these processes not only result in low oxygen, it also accumulate carbon dioxide in the coastal estuarian region. And this is a paper, this are figures from Professor Wei Chai, and he summarized this, the study of coastal ocean acidification. And we also see a similar phenomenon in our study is that the PCO2, the carbon dioxide mix, concentration in the surface water is extremely high in the estuary, and the pH value is lower and lower. And meanwhile, there's also because of the hypoxia, which can produce more greenhouse gases such as nitrous oxides and methane, and these make they contribute to the global warming and make the situation even worse. So yeah, that's some backgrounds of of this study. And what we are doing now is for the past uh, decades, we keep studying the carbon dioxide and the greenhouse gases, including methane and nitrous oxide, and we study their impacts on the um, marine environment, so-called the ocean acidification, and we study the aerosol because they play a negative role in the global warming. Then we try to develop our technique to promote our study. The study area covered the Southern Ocean and Arctic Ocean, and also more recently we begin to focus more on coastal region and estuary. So here is some result of our study. This is a study about the Arctic Ocean acidification, and we see the expansion of corrosive low pH waters in Western Arctic Ocean in the past 20 years. This increasing is fast and shows that Arctic Ocean is acidified quickly. And this work is published on 2017 on natural climate change. And there's also some work about the polar region, but I don't think it's the work I should present here. So maybe next time. So about the coastal and estuary study, I think that's something we are recently focused, and we think that's very important. And the study site include Jiulong River Estuary, which is nearby Xiamen, and also Beihai, Guangxi province, China, which we studied once or twice about a mangrove. And we have a collaboration with Jiulong University on river estuary and the coastal sea ocean acidification. And research, the collaboration is now, because of the pandemic, there's a somewhat postponed, but we will keep doing that. Okay, so for the Jiulong Estuary study, this river is in Fujian province, is the second largest river, and it's heavily polluted by nutrient discharge, and mainly because of the fertilizer use and livestock waste discharge. So we see that we can see that the carbon dioxide surface carbon dioxide partial pressure can be as high about 3,000 micro ATM. That is extremely high value, and correspond to this high PCO2 value, we see a low pH value, which can be as low as about 7.0, something like that. And correspond to this, we see the dissolved oxygen saturation anomaly is the dissolved oxygen saturation of about 40%. So that means with the high concentration of nutrient and lower concentration of dissolved oxygen present, this environment, this water tend to be a very acidified water mass, and yeah, we think that that's because of the eutrophication contribute to this acidifying. Okay, and we also collaborate with Jiulong University about the carbonate chemistry near the Malaysia coastal water. On the technique development, and we try to develop some automatic instrument. This one is developed by our group. It's a greenhouse gases underway automatic one, automatic underway system for surface water and atmospheric greenhouse gases measurement. And this system was just deployed on board Follow Stern for mosaic prediction. It work continuously for almost 1 year. And some other instrument we developed by ourselves is the underway system for DMS study, which is also deployed on board a South Africa icebreaker for the Southern Ocean study. So I think let's talk about it. Dissolving organic carbon measurement. That's the main point of this presentation, and try to develop this dissolving organic carbon technique, which can measure the surface water continuously along the cruise track, or you can use it on a time series station. So I will give more detailed introduction about this system. So we know that to measure the DIC, generally there are 3 steps. First, you get a water sample, and you convert the carbonate or bicarbonate species to carbon dioxide by acidifying the water sample. Then the next step is you using the gas diffusion approach. Yeah, sometimes it's just bubbling, and sometimes maybe using some membrane to separate them. You can separate the carbon dioxide, and the final step is that you measure the carbon dioxide or the carbonate using different methods. In our labs, we use non-dispersive infrared. I think this is a very common method. More recently, the calibrated ring down spectrometry, which has lower detect limit, and there's also a very high resolution methods, but the instrument is very expensive. And also there's a spectral photometry methods. All these methods used to measure carbon dioxide release from the sample. And there's also another method which use the electronic conductivity, and the contact is captively coupled conductivity detection, which is we also call it C4D, as measured the carbonate. In this method, we use this electronic conductivity detector, and yeah, this is the 2 methods I mentioned. The one is we use in our lab. The other one is also common. They deploy this on board different research vessels. And for our method, this is the scheme of the diagram of this instrument. So the standard solution and the sample can be injected into this system by injector, and it goes through the sample loop. And when they fill the sample loops, the valve will switch, and the acid goes in to mix with the sample and go into a gas diffuse assembly, where the carbon dioxide would diffuse through a membrane, and on the other end, it mixes with the molecule water, which the carbon dioxide in which is already removed, and the diffused carbon dioxide dissolved in this carbon dioxide free molecule waters and go into the detector, and then we finish this measurement. So with this methods, we can have a link. This detector has a linear range of response from 0.1 to 4 micromole per liter, and the limits of the detection is about 0.5 0.16 micromole per liter, and the throughput of the sample is about 12 samples per hour. But this could be improved if we have more injectors, or we can do some upgrade about the system. Okay, so here are just some diagrams of the results of this system. So as I showed before, there's a good linear range of 0.1 to 4 micromole. So here we can see the green line. But beyond that, the curve is non-linear response, but it's still it fits well with the R-square of 0.999, and yeah, we think that's good. It's a very good fitting curve, and concentration is stable in 2 hours. I think the RSD of the repeatability is about 0.56%. So this is a very stable instrument. And when we compare the data of this method with that derived from the C4D detector, they agreed with each other well. So yeah, here is a figures of field work in Jiulong Estuary when the research was stopped by the dock, and the green line, the green dot is the underway measurement results. And we can see that the variation of the DIC concentrations consists well with the variation of the salinity, or you can see the tidal cycle. And the green lines and the green dot and the red dot agree with each other. And the red dot is the discrete sample measurement result. But it's a pity that the pump of the ship stopped here, so we don't get any data here. But anyway, we think that this instrument, the fieldwork of this instrument is also good. So we are also analysis the water sample from different depths, from 0.5 meters, 3 meters, and 5 meters, and we see that the highest DIC concentration will present in the depths of about 5 meters. So in the bottoms of the estuary water, the dissolving organic carbon concentration is higher. Maybe because there's an emission of these carbonates from the sediment. Okay, so the results already published, and here's if anyone wants this paper, I can forward you. Here's the summaries of this work. So first, this microvolume flow with 100 microliter was achieved in this study. And so for this instrument, we only did AC carrier stream and ultra pure water stream to measure this to measure the DIC, and we don't need any addition indicator. And the second is the DIC observation system is sufficient for underway observation of marine DIC, and which can provide high spatial and temporal resolution data for DIC concentration. And the third, the DIC concentration profile reflect the whole tidal cycle well, indicating that the time solution of the underway DIC observation instrument is sufficient for the estuary water study. So that's pretty much of the method itself. And the last part of the presentation, I will show you our plan in the future. So for the technique, we are already developed some technique for the ocean acidification study, and now we are upgrade this DIC methods to its version 2.0, and next step we will try to integrate it with a high precision pH sensor, temperature sensor, and salinity sensor. And we already upgrade this software. And after this, and we will integrate it with also a total alkalinity underway system, and together make them a whole system for underway observation of the carbonate system parameters. And for the greenhouse gases, and we will collaborate with Chinese Academy of Sciences they already developed these greenhouse gases laser analyzer. So we can use this system for coastal region study. And for the study side, Jiulong Estuary, yeah, because it's just nearby, it's very close to our lab. So, and it has a serious environment problem. And we think that's a good site for us to study. And indeed, for the past 10 years, the government already make large efforts to try to recover the environment of this estuary. And the action including the downsides of the poultry breeding and livestock feeding, and also the next step there will be a waste treatment of this livestock and poultry breeding discharge. And the coastal agriculture was banned since 2009, sometime, I don't remember, but anyway, now there's no agriculture. And the next step also including the organic fertilizer use instead of the industrial manual ones. Yeah, I think I recently I just read some paper about this. That's the plan of next step of the government. So yeah, I think the environment indeed is already the environmental history is already improved a lot. However, here is still some of our results shows that the dissolved in organic carbon, that dissolve the nutrient, the concentrations of nutrient in the past 10 years. Indeed, it already reached its maximum 10 years ago. So since 2010, the concentration of the nutrient, nitrogen-based nutrient is relatively stable. They don't increase significantly anymore. However, we can see that the greenhouse gases concentration is keep rising. So even there's a rise, you can see that between 2012 and 2018, the concentration of the greenhouse gases increased 1 order of magnitude higher. So that's very surprising result, and we think that we don't have the data, the carbonate system data of that time, but we think it's something that we should taking care of, because we think that the nutrient don't increase, but the greenhouse gas concentration increase means that maybe there's something happened somewhere. And based on our result in the underground water, we see that there's a high concentration of greenhouse gases in the underground water. And we believe that there will be a carbon dioxide enriched water too. So yeah, I think here underground water is a environment that we should not ignore. So for the study site, and we will keep doing this, I will keep studying the Jiulong Estuary. We will use, including the cruise, we will use the land-based time series station. We will study the underground water, and we will study the rich nutrient loading of this river. We will see where is the hypoxia exists, the serious hypoxia exists in this estuary, and we'll see the carbon chemistry. We'll study the carbon system there to see the progress of this ocean acidification in this estuary. And we also study the greenhouse gases aerosol. For aerosol study, that we study aerosol because we already know that in the coastal region, maybe the nutrient in the water, they're about 30 to 40% of nutrients contributed by the aerosol transport and deposition. So to study all this, to see all this environmental condition, we can find the where is the problem is, and we can work together with the policymaker to make this estuary, the environmental quality of this estuary better. So for collaboration in the future, now we already have a plan of joint lab studies in Malaysia, Terengganu University, and because of the pandemic, we plan to send our instrument there to contribute to work together as a lab to monitor the ocean acidification in Bidong Island. Unfortunately, because of the pandemic, our instrument is not able to send there. So in the next month, we don't know when, but somehow when the system, when the instrument is ready, when the pandemic is over, we can finally send this instrument there, and we will monitoring the parameter, the carbonate system parameter there, together with the standard methods for this ocean acidification study. And I think by this way we can exchange the low-cost technology. We can share this technology with the collaborator. We can promote this research work together. So yeah, pretty much that's our work, my presentation, and we're working on collaboration and looking for more collaboration opportunity chance. Yeah, that's all. Thank you very much.
Yeah, thank you very much, Dr. Li Yang. I think it's a nice presentation, and then we congratulate for your progress of develop of the underway measurement of the DIC, and are looking forward, to work with you in the near futures. For the question for the Li Yang will be, in the end of the presentation session after the third speakers, right? Thank you very much, Li Yang. We are going to the second speaker. It is Dr. Cheromrat Sangmani. His background is physical oceanographer. Currently, he's a researcher at Phuket Marine Biological Centers, Department of Marine and Coastal Resources in Thailand. He has leading an ocean acidification monitoring research programs of the Department of Marine and Coastal Resources during the last several years. He also involved in the coastal water quality monitoring program and research on ocean dynamic and processes, particularly in the Andaman Sea. He's going to give the talk today on the ongoing observation of the ocean acidification in the coastal water in Thailand, which is a part of the WESTPAC OA monitoring programs. The progress and result will be presented. Dr. Cheromrat, please, the floor is yours. You can share your screen. Yeah?
Thank you very much, Professor. Let me share the screen. Can you guys see my screen?
Yes, we see.
Okay. Let me introduce myself again. My name is Charamrat Sengmani. I am the researcher of the Department of Marine Coastal Resources, Thailand. For today, I would like to share with you our work in the Department of Marine Coastal Resources. The content.
Charamrat, could you share the full screen?
The full screen? Oh, okay. Yeah. For today's talk, I will present about who we are, I mean the Department of Marine Coastal Resources, and why we work with this project, and also the project aim and methods, and also some example of the result that we obtained during the project. Who we are. The Department of Marine and Coastal Resources is an governmental agency that's responsible for the protecting marine environment and also coastal resources like coral reef or mangrove or fish, something like that, and also to make it to do the conservation of all the environment. Yeah, I mean the marine environment. So with that, one of our mission is that to provide a guidance for consideration of the policy for the management, conservation, and restoration of the marine coastal resources. So surely, that guidance must be based on the research and development of any technique or even the innovation that we try to serve our mission, to serve strategic purpose. And how do we involve with this project? As you can see that under the climate change, oceanification is one of the global concerns that could bring about the harm to the coastal environment, coastal resources. Why I say that is global concern? Because you can see from the Sustainable Development Goal that was put into the goal 14 number, the target 14.3, which is to minimize and address the impact of oceanification, including through the enhanced scientific cooperation at all levels. With that indicate one of the indicator associated indicator to the target is to have the average marine acidity measurement at the representative sampling station. So because as our main mission to the protection of the marine environment, that's how we involve with this with this project. But how to achieve in the project? Under the program of IOC-WESTPAC and the support from the DMCR, this project, the Ocean Acidification Project, has been initiated and run by the Marine and Coastal Resources Research, Development and Institute under the DMCR. The project aims for two parts. First of all, we try to address the pH in the Thai water. The question to be addressed about this part is that what is the current state of the pH, and also in which direction that we are heading to. Another part is to assess impact on marine ecosystem like a coral reef. We try to get the idea how bad oceanification can make harm to the coastal resources. So to achieve the purpose, one of the steps that necessary for us is to have the capacity development with our staff. So convened by the IOC-WESTPAC, 5 training workshops were organized in the Phuket Marine Biological Center in order to build up the transfer necessary knowledge among the member countries. As I said that two parts that we are aiming to is to try to get the idea of the pH, current pH in the ocean, and also to assess the impact. Spectrophotometric pH measurement method, and also autonomous reef monitoring structure, or ARM, was this kind of knowledge was transferred in the training. So let's move to the first part. We try to address the pH in Thai water. So with that purpose, we set up more than 100 stations to monitor regularly monitor the pH in the water, in the surface water. You can see that include both the Gulf Thailand and also Andaman Sea. In the Gulf Thailand, you may see the station along the coast of the eastern part of the Gulf Thailand, or even in the upper Gulf Thailand. And also in the low part of the Gulf of Thailand, you can see so many stations, both in the coastal area, also go offshore in the more deep water, in the deeper water. And also in the Andaman Sea, you can see so many stations, both in the coral reef area, also in the open area. This I just like to share you about the activity that we did in the field. When we go to the sea, we have the environmental condition, I mean the temperature and salinity measured by using the conductivity temperature depth instrument. Also, we collect the water sample to bring it back to the laboratory. As for the method that we use to determine the pH and the total alkalinity, for the pH measurement, we use the spectrophotometric method in order to do that. And also for the total alkalinity determination, we use the auto titrator in order to find out how much the alkalinity in the water sample. With these almost, let's say, 5 years observation, quite a long time that we really go to the sea and collect the water sample. This is just some example of the result that we obtain. You can see this is the result from the south of Phuket Island, including the Racha Island, Rachayai and Rachanoi, Maiton Island and Hei Island. You can see so many measurement in the picture. For the left panel, the upper left panel is the pH measurement, which is up to now with the 5 observation, we can say that the mean state of the pH is about 7.9, 7 around that. And the trend is just like, because it's still a short-term observation. This probably due to the climate variability in a short term, in a short run. But the trend with this short term is say that is about, this area is kind of increasing, just very little, 0.003. And also the total alkalinity is about 2,100. But with these two parameters, we can also have an estimate of the so-called aragonite saturation, which is kind of one of the indicator to the coral reef in order to understand if the impact on coral reef gonna be high or low. But that's just based on the chemical parameter. Another example of the observation is in the upper Gulf Thailand. You can see from the picture that for the upper Gulf Thailand, the mean state is about, with the observation, the mean state is about 7.94, but the trend is going down quite a little bit. And also total alkalinity is about 2,000 also. But with this, the estimate of aragonite saturation is about 2.9. The question to be mentioned about is that, is it, for this level of the aragonite saturation, is it sufficient to maintain coral reef in the future? This also some kind of the result, because it's not only that we collect the water sample from the surface, we also collect the water sample from within the water column in order to understand the contribution, I mean the influence, what may cause the variation in the pH. For the left picture, you can see that we collect the water sample in the upper Gulf Thailand. That's what is the eastern part of the upper Gulf Thailand. You can see from the pattern that for the top one, the color represents the salinity of the water obtained by using the CTD. And also the lower one, you can see the color represents the pH measurement. Also the contour show the disoxygen. That's from the 3D. But when you see it, you can see obviously that the resemble between both parameters is quite resemblant. In other words, they kind of similar. And somehow with this, you can say that freshwater may have some contribution or influence the change in the pH variation. That might be through the kind of productivity or whatever. But in the Andaman Sea, you can see that if you collect the water sample from the very deep water, about 70 or 60 meter deep, you can see that the pH go down from the about 7.9 or almost 8 near the surface to about 7.8. Which is this kind of, you can obviously say that it's kind of natural cause because the intrusion of the deep water in the bottom layer. This kind of to try to understand what's behind the change. Not only try to get to the idea of the variation of the pH, we also have duty to submit the data to the SDG because we are government, we are in the government, right? With this, we also submit the data to the SDG 14.3.1 data portal that's developed by the IODE. And we also regularly report the pH measurement to the SDG annually, yearly, yeah. Then we come to the second part, which is to try to assess the impact on marine ecosystem, which is actually we focus on the coral reef. For this slide, I just like to show you the chronology for the study of autonomous reef monitoring structure. The method that we use for the study. From the chronology, you can see that we have during the training, during the workshop training, we have the knowledge transfer from the NOAA under the great help of the WESTPAC program. So in 2016, we have the arm deployment around the Phuket, in the Panwa Cave, Phuket. And then 1 year later, we have the training about how to recover the arms. Also, how we can correct the specimens from the arm. And also in 2018, we extend our station to all around the coastal water in Thailand. And recently, we have corrected. In 2019, we recovered the arm already. Yeah, this just like to show you about the deployment of the arm in the Thai water. Right, just a short video. This is just some activity that we did. Let me accelerate a little bit. Right, this kind of the way to recover the material. Then this picture I would like to show you about after a year of deployment as the Panwa Cave. We have the training. Under training that's organized by the WESTPAC, we have the train of how to collect the specimen from the arm. That's all for the impact part. But the thing to think about as a governmental agency is that how we can deliver our message to the policymaker, which is the main purpose of any work of us. So this kind of the simple example, very simple example that we may think of, like we try to identify the impact area for the acidification, because in the project we get the pH trend. We can estimate the aragonite saturation, which is the state that which is one of the indicator to the coral reef, and we get to the idea of the how worse impact can be. But that for the biodiversity, I mean for the arm is still in process. But let's say for just 2 parameters, we can see that for the pH trend in the upper Gulf Thailand, you have the decreasing trend, and also the aragonite saturation is the lowest area of the aragonite saturation in the Thai water. With this, but in the Andaman Sea probably is quite fair. With this, you can identify which area that probably we need to take care of or we think about it. Including that, if we know the impact, as I mentioned in earlier, that freshwater have a kind of influence to the variation of the pH in the Gulf Thailand. We may think about the policy that must be implemented in order to try to get to the better situation. But it's just some very simple example of analysis that we think about. Then, because of for the next decade, we are moving forward to the decade of action. That's called for acceleration of sustainable solution to all the world biggest challenge. What to think about probably somehow what's the better message to be delivered to the group of policy makers. In my opinion, something like some funny thing that we may think about is that having livelihood adapted to suit the changing environment, actually degrading environment or preventing the environment from changing to maintain our current livelihood. Which option is the better option? That's kind of the thing to think about in the next decade, probably. Thank you very much. That's all my presentation. I will return the stage to the Professor Somkiat.
Thank you very much, Lamlat, for your good information. From what you have been done doing several years, and what you are going to promote your work to the policy peoples. And the next, I would like to go to the next presentation is Dr. Wa Ibas. She has a background is in the field of the biology and environmental science, and has a long experience on marine algae and aquaculture research. Currently, she is a senior researcher and associate professor at University of Palu Orios, Indonesia. Today, she will give her talk on the effect of the ocean acidification on growth lipids and carotenoid contents of the widely used green macroalgae in aquaculture, Calelas vulgaris. Please, Dr. Voss.
Okay. I'm trying to fix my scarf, but I don't know. Okay, thank you, Professor Somkiat and other presenter for the opportunity. I'm very grateful to be able to participate in this discussion about ocean acidification. So my talk today will be a little bit different from the previous two speakers that already talked about the ecological and technical aspect of the ocean acidification. I guess what's happened with my scarf. Okay. So my talk will be in this flow. First, I will talk about the microalgae, and then the lipid and carotenoid production in microalgae, and then possible effect of ocean acidification on lipid and carotenoid content of Chlorella vulgaris. And then the last part of my talk will be about the research question, actually, and what we can do to use microalgae as bioindicator, and also maybe some key species in detecting ocean acidification. So for the introduction, I will talk about briefly about microalgae. I expect that most of the audience already know, or at least a little bit familiar about microalgae. They are a microscopic single or multi-cell eukaryotic, some also prokaryotic algae. The term of algae is encompass the whole eukaryotic plant in aquatic ecosystem. And because they are eukaryotic and also photosynthetic plant, so they are the basis of aquatic food web in all aquatic ecosystem, and they play very important roles in the ocean. Not only for the basis of aquatic food web, but also their role is increasing for carbon sink in the face of ocean acidification and climate change. So, yeah, so because they are unicellular and microscopic, of course, they can only be seen under microscope. Sometimes you can see it with low magnification, but to identify the family, we have to see them under 100 or up to 400 times magnification. Reproduction is by cell division, and some species have on movement because they have flagella, but most of them is drift passively with the water current. So far, the biologists divided them into 9 divisions according to the pigment types. And like I said before, they are photoautotrophic, but some also heterotrophic organism. This is, I just will skip this very fast. This is the division of microalgae, and so 9 division, and their habitat. So like I said before, they are found in all aquatic ecosystem, in all, almost in all habitat, even in the terrestrial, and also living in symbiotic. They are a very important symbiont with coral reef, forming what we call it now zooxanthella. This is the main pigment. This is what I interesting to in my research, in my current and future research, about harvesting the carotenoid content from microalgae to be using as antioxidant for functional food, but also for aqua feed for aquaculture industries. So Chlorella vulgaris, the species that I'm interesting to study belong to chlorophyta. So they have alpha, beta, and gamma carotene. So this is the roadmap of microalgae biotechnology research at my university. So we're gonna look at from 2018 to 2025. So I'm also interesting or developing. Now my student is currently working to observe the growth and the biomass productivity of Corella, Corella vulgaris under low pH. So we hope to use microalgae as a bioindicator of climate change, particularly the effect of lowering pH in the culture condition. So this is all that we already do. We already did like a lot of research, like to optimize the culture condition, and also searching for organic nutrient to be used in microalgae culture. So why? So that's from ecological standpoint, but also from biotechnological standpoint. Thus, the commercialization of microalgae is a very lucrative industries to be developed in. Maybe this is not common now in Southeast Asia, but like in Australia, and I know in South Korea, and maybe also in China, the microalgae industries for several purposes like nutraceuticals, food and antioxidant, and now the attention is being drawn to use algae or including microalgae as bioenergy. So this is why maybe the ocean acidification will threat the development of microalgae industries. Because like right now, we use most of culture media using marine or natural seawater. So what's going to happen if in 15 or maybe 100 years from now, if the seawater pH is decreased? Whether is it going to make the microalgae industry better, or maybe it will crash, and thus this will impact the economic side of the industry. So this is the value of the product, microalgae product. So for antioxidant and food, it possess a higher value compared to bioenergy or animal feed aquaculture. But the market volume, so it will affect the market volume. The market volume for nutraceutical for the antioxidant is what you call it? It's small compared to bioenergy or biofuel. So yeah, because of the high price and high value of antioxidant, food and nutraceutical, this is, and also from pigment and omega-3 fatty acid from microalgae. So that's why I'm interested to produce lipid and carotenoid from microalgae using, so far now I'm using the organic culture media. So how the lipid and carotenoid produced in microalgae? Research, a lot of research shows that lipid and carotenoid is produced under stress condition, whether it's nitrogen or phosphate or sulfur. This is a nutrient stress or light temperature salinity. But very little study is conducted to see the effect of low pH or changing in pH value in the culture water. So this is the background or the basis of my research. And then by using the stress condition, we can then produce biomass and high lipid and carotenoid content. Usually they use two-stage cultivation strategy, but maybe this is not so what you call it. It's commercial. It's not really favorable in commercial condition because it's quite expensive and then it's time consuming. So the research now is how to do just one stage or one stage culture strategy using stress condition, but then we can harvest a high lipid content, high lipid and carotenoid content, but also high biomass. We can do also genetic engineering by producing transgenic microalgae, but then it needs like some previous or background studies about genomics, transcriptomics, or metabolomics. So like I said before, like the previous slide, light stress, nutrient stress, and salinity stress. This is the common precursor for producing microalgae pigments. But again, we don't know yet. We don't know how the pH will or the concentration of dissolved inorganic carbon in culture of microalgae does the low pH will affect the pigment production and the growth of microalgae. This is how carotenoid is producing in within cellular level. So using Porphyridium species, this is the orange border, to produce carotene, and then Tetraselmis to produce lutein, and Haematophoccus, which contain astaxanthin. Each pathway is different, and then the end product also different. So this is the research that already done by Kamarsaini et al. This is, I think, 2019 or 2020, to see how the high or light stress or nutrient stress, both deficiency or sufficiency, affect the production of beta carotene or carotenoid in a cellular level. So how beta carotene production is actually regulated by gene. So different gene is responsible for different pigment. That's the point that I want to make. So beta carotene, astaxanthin, and lutein production are producing by expressing different gene in microalgae. So this is also some area that we're going to see how pH stress will affect the production of this kind of pigment. So this is what we already did at my lab at UHO. So this is the species. This is 10 times magnification. Like I said before, you can see under low magnification, but the cell is quite clear, but then you cannot see the whole structure of the cell unless you increase the magnification up to 400 or 100 to 400 times. So this species is widely used for live feed in aquaculture, but also widely produced for functional food because they contain high protein and also high omega-3 fatty acids. We have been maintaining this species more than 2 years in my lab, and we use it as a model culture for media optimization and also culture condition optimization. We also show that increasing salinity up to 40 PSU affected the growth and final cells density of this species, but not the biomass productivity. So it means that at all salinity tested, the biomass productivity is similar, but the growth rate is different. So this is the study that we already did to show that nutrient deficiency can affect the content of carotenoid in Corella vulgaris. So this is, we use the fermented water hyacinth, and this is the nitrogen and phosphor concentration. Compared to F2, the widely used culture media, the content of nitrogen and phosphor in water hyacinth is higher in all concentration tested. So this is like nutrient, not deficiency, but maybe sufficiency, like above the required by the macroalgae. So we can see here the growth curve is similar to F2. Some of the concentration show similar, but at 1% of water hyacinth, we can show the growth is very, they reach the dead phase faster than other concentration. But this is the specific growth rate or the for each concentration tested compared to F2. So the 1% concentration show higher growth rate compared to others, like you can see from this graph. This is the biomass productivity and carotenoid. This is what we very interesting finding, that biomass productivity always what you call it, it's not linear with the carotenoid content. So the higher biomass productivity, usually the lower carotenoid content. So this is also confirmed by other studies that good growth not necessarily show the good pigment or the good antioxidant content. So this is partly because carotenoid is antioxidant produced by microalgae to prevent their cell from damaging environment or from stress. So the more stressful they are, the more they produce carotenoid. So growth is an indicator. We want to achieve optimal growth with optimal carotenoid. That's the thing that we're gonna, because when you sell the microalgae, you sell the biomass actually. So we want to sell the biomass, the sufficient biomass with high carotenoid content. And this is the lipid content of microalgae under sufficient or over sufficient nutrient. So high lipid content was found in 5% media. This is high nutrient content compared to other concentration tested. So lipid is important for biofuel or bioenergy, but also for omega-3 fatty acid. So this is the future studies that we're gonna see. What is the effect, the possible effect of ocean acidification on lipid and carotenoid content in Chlorella vulgaris. So I don't want to speak for this slide because the previous speaker already explained what is ocean acidification related to warming and deoxygenation. So the studies by Jean et al. And Gao et al. Show that diatom, another class of microalgae, this is silica microalgae that contain silica. So the high carbon dioxide concentration, the show in red line. So the growth rate at higher carbon dioxide concentration is lower than low carbon dioxide concentration in blue lines. So so this show that at higher concentration of carbon dioxide, which means that the pH is lower, the growth rate is decreasing. This is for diatom. But for coccolithophore, the trend is the reverse. So at higher carbon dioxide concentration, the growth is increasing compared to low carbon dioxide concentration. What about in green microalgae where Chlorella is belong to? We don't know yet, but the study with Arctic Chlorella species show similar trend with coccolithophore. They show that at higher carbon concentration in culture water, in culture condition, Chlorella species from Arctic, the isolated Chlorella from Arctic, show a higher growth rate compared to when they grow under low carbon dioxide concentration. So this is interesting because it means that low pH, high carbon dioxide concentration in water will stimulate the growth of green microalgae, but not diatom. So maybe from ecological standpoint, it means that they're gonna shift the structure of microalgae community in the ocean. But what about in culture condition? And for biotechnological standpoint, that was actually a good news because when you grow microalgae under high carbon dioxide, high dissolved in organic carbon dioxide, which means at a lower pH, the growth of green microalgae, include Chlorella, may not be what you call it, decreasing. So from biotechnology, this is beneficial. So, but what about the other metabolite in the microalgae? So talking about the bio indicator, if we want to see whether the microalgae affected by ocean acidification or not, we can use actually one of important fatty acid for biofuel, which is palmitic acid. Palmitic acid usually found in other plants that will be used for biofuel. So this is some metabolite that we're gonna have to investigate if you want to see whether a microalgae was affected by whether pH affected the growth or the lipid content of microalgae. So way forward, we want to investigate what is the effect of ocean acidification in Corella vulgaris culture. Whether it's increasing or reducing the photosynthesis, that's the growth, or if they are increasing or reducing the biomass. If growth of biomass increasing, lipid and carotenoid might be decreasing, not an optimal scenario for commercial production. If growth and biomass decreasing, lipid and carotenoid might be increasing, but this is not also an optimal scenario for commercial production. We want biomass, lipid, and carotenoid at optimal level for optimal commercial production. So more studies will need to address the metabolomics of microalgae and what is the effect of ocean acidification on microalgae in control condition. So, and then the last question that also interesting to investigate, it will ocean acidification affect the sustainability of microalgae industry? And what about other related industries, such as aquaculture? Because microalgae is widely used as natural feed for aquaculture species larvae in hatchery. So I think that's all my presentation today. Thank you very much for your attention, and I will be happy to take questions. Thank you very much.
Thank you very much, iBoss. I think it's now we time for the open the floor for the questions. If any of you would like to ask the question, please raise the hands. And if you need for specific speaker, please tell the names, and then I will. You can ask the question directly to those people. Yes. Now I saw the Li Yang. You raise your hand, right?
No, I just grabbed my hand.
Okay. Yes, then we have one Kanita. Yes, it's time for you to ask the question.
Thank you so much, Dr. Somkiat. May I introduce myself? My name is Kanita Uthaipan. Do you hear me?
Yes. Yes.
Thank you. I want to ask Dr. Li Yang 老师, from your system, you investigate DIC in the estuary. So in the estuary, in the zone of the high turbidity and turbulence that is driven by tidal. So my question that is, the turbidity and tidal turbulence, is it affected on the DIC on the autonomous system or not? And how you fix it, the problem from this one? Thank you. This is my question.
Yeah, Li Yang, you can respond to the question.
Excuse me, I didn't catch up with that question. Would you please repeat again?
Oh, okay. I will go slowly. Sorry. In the estuary, it is quite high turbidity and turbulence.
Oh, okay.
So I would like to ask you the autonomous DIC system of you that it had affected the turbidity affected on the system or not?
Yeah, sure. I think the turbidity will it will influence the measurement because the tubing is a small one. I think it's 1/8 inch, 1/8 inch, something like that. So before that, we need to pre-filter the inflow water using them, some kind of filter to filtrate the particulate matter, to remove those particulate matter before measurement. Yeah, that's it.
Yeah,
thank you so
yeah.
much.
Thank you, Li Yang. Is the next one Dr. Fuji, right? Yeah, it's a for you to ask the question.
Yes. So I'd like to. Do you hear me?
Yes, yes.
Yes. So I'd like to ask to the Dr. Eva. Dr. Eva.
Mm,
Do you hear me?
Dr. Eva, you still there? Yeah.
Yeah. Hello. So I have
Yes,
two.
I heard.
Yeah. I have the two questions. So the one is, are you going to do the experiment by the raising phytoplankton in a high CO2 the conditions? And the other is, so that you mentioned about the cocolis flare can be the positively affected by the high CO2, but the shell can be the negatively affected because they make the shell calcium carbonate shell. So I don't think ocean acidification can affect in both ways for the species.
Yeah, Dr. Eva, you can respond to the question.
Yeah, if I hope I'm not mistaken, but what I understand from the study, that was not my study actually. That's from other studies. So they don't talk about the cells per se or the calcium carbonate in the cell, but they look at overall growth of lithophore under high carbon dioxide condition. So because that was not my study, I cannot answer that very comprehensive. But what I can see from the paper is that they only investigate the growth of coccolithophore. They're not talking about the calcium carbonate. But that's, I agree with you that coccolithophore produce carbonate in their cells, and maybe in the long term, their growth will be declined because of that. Because of if the higher carbon dioxide condition is prolonged, maybe the growth will be impaired. So yeah, that's all that I can answer from that paper.
Yeah, so thank you very much. And then how about the other question? So are you going to do the high CO2 experiment? So raising CO2 and then culture.
Yeah. So now my student in the lab still working on that. They are culturing Chlorella vulgaris under low pH condition. And then at the end of the experiment, I don't know, maybe the next 2 weeks or 3 weeks, we're gonna see whether the growth will be affected or not by the lowering the pH level in the culture.
Yeah, terima kasih.
Yeah, thank you very much for you. Yeah. So any more questions? If you have the question or you have any suggestion or recommendation about the OA research or the work in the future, also is welcome. Yeah. No more?
Somkiat, may I ask a question?
Yes, Maria, it's okay. Yeah, for you.
For Dr. Iba, may I ask how you change the pH? Do you bubble your system with CO2 or you just adjust the pH by adding acid or base?
Yeah. Thank you for the question. I don't use the carbon dioxide bubble to decrease the pH. So we just use the acid to decrease the pH. So yeah.
Yeah, I think it will make a difference if you use CO2 because you're dealing with the photosynthesis part of the plant. So intuitively, with higher CO2, you induce more photosynthetic reaction. But I don't know if that's the same effect with just manipulating the H plus ions.
Yeah. The thing is, it's very difficult to find the CO2 here in the eastern part of Indonesia. So that's also quite, it's not about the price per se, but the supply of CO2 gas. But yeah, I know that's the very, very good input. And then we're gonna see if in the future we can also use the CO2 bubble to decrease the pH. Yeah, thank you for your insight.
Thank you, Maria. Yeah. Next one, the question. Chawalis, please, the floor you. Open your mic. You mute your mic, the microphone. Turn on your microphone.
Got it. Sorry about that. So Chawalit from Chulalongkorn University here in Bangkok. I've got a question for Chalumrat. If he's still around.
Yeah, she should be around. Yes.
So let me just state my question right now then. It's a nice trend that you were showing in your slides. And I was wondering whether you have any theory as to what differences you see from, you can see the trend that is so different, like pH trend in the upper Gulf of Thailand versus the mid Gulf and lower Gulf and the Andaman Sea. What could potentially attribute or causes the such differences? Especially you see that there's clearly the trend of pH decrease in the upper Gulf of Thailand as opposed to the other area in your study. Thank you.
Thank you for your question. For the trend, let's say first that because it's still 5 years observation, so it's still a long time for us to estimate the exact trend in climate time period. But as I showed you in the slide, that when we go through some kind of we collect the water sample. I mean, we collect the water sample in the water column. We can see the difference in the pattern or little waves influence that could probably affect the variation in the pH in the water. In the trend, we just use the pH measurement from the surface water. I mean, the 1 meter below the surface. So that could probably related to some kind of for the upper Gulf of Thailand. Just my guess. That could probably related to the fresh water or some kind of variability in the climate, but also can attribute to the kind of the activity like productivity or nutrient or whatever. I mean that can affect the change in the pH in the surface area, in the surface layer. Does that answer your question?
I guess so. And I have another follow-up question. As we all see in the first speaker, do do you, as part of the DMCR, plan to have any kind of continuous measurement, like like what the first speaker was presenting, or anything like that?
You asking me, right?
Yes.
Yeah. For the plans for this project is still ongoing. As I said that when we talk about the climate change, it's kind of the it's kind of the climate time scale which is far longer than 5 years. That's the thing that why we still continue our work. But other than that, we try to get to the assessment of the impact of the land influence, like to the OA. You can see that we try to extend our area of study into the kind of the so-called hypoxia that kind of serious in the Africa of Thailand. And then with the technique that developed based on the acidification project, then we probably step forward to also tackle the problem also. Thank you.
Thank you, Shramrat. Yeah. Yeah. Any suggestion also welcome, or even recommendation to, the direction for the WESTPAC to go forward from your from the speaker today. So it seems to be no more questions. Yeah, I think I thank you very much for all of you to participate in the meeting today. And you know that we still have the 3 more webinars in the next week, and then the week later, the next week will be quite interesting because we are going to talking about more related to the corals, because it's very important to our region, or even to most of the country in the Southeast Asia, and then even in the southern part of Japan or China as well. I hope that I would like to invite you to join us again next week, for the webinars. And then if you by using, you can use the same link as you have today, right? And next week the moderator will be Dr. Li Yang, and then we are going to continue our webinar. If you have anything in your mind about to develop this kind, the work or study on the impact of the OA on marine habitat or environment, you can make the brainstorm during the discussion as well. Yeah, maybe this is the first one. Maybe that we are still new with the presentation and then discussion, and then the next one, you are welcome to give any suggestion and recommendation. Thank you very much, and looking forward to see you again next week. Please join us. Yeah.